Now, welcome to our today's science lesson, standard 8. That is still on the topic plants, but now we are going to discuss how fertilization in these plants occurs. During our last lesson, we talked about reproduction, the reproduction parts of the plant, and we discussed about the parts of the plants and where the male and the female parts of the plants. We gave the name that is given to the female part of the plant, and also we gave out the name that is given to the male part of the plant. Now, after you understand the male and the female part of the plant, now we come to discuss now how do they unite or how do they join so that now they can carry out fertilization to produce their fruits and the seeds. Now, straight away, we are going to discuss fertilization in plants. And now, before we discuss fertilization, you have, first of all, have to get the understanding of the term fertilization. Just like we discussed in animals, where we talked about fertilization being the union of the male and the female sex cells. But now here, there is different parts that are now involved in this fertilization. Now, when you talk about fertilization in plants, it occurs in the ovary. The fertilization in plants occurs in the ovary. Now that is the female, the male part of the plant, that is the pollen grain, which is the male sex cells. Now they fuse with the female sex cells, and we say the female sex cells in the plant is the ovules. We have the pollen grain as the male sex cells, then we have the ovules as the female sex cells. Now the two unite, or they join each other in the ovary. Now when they join each other or unite, that is what we define as fertilization. Now after understanding that, we move to what now happens so that now we can have fertilization. Fertilization in plants occurs after we have a successful pollination. We discussed about pollination where we gave out the examples of pollination, that is the types, that is the self-pollination and the cross-pollination. Now what happened immediately after we have a successful pollination? When successful pollination occurs, there is germination of the pollen tube that occurs at the stigma. Now we discussed the flower, that is the flower part. This is the female. Now this part here is what we call the stigma. Now after the pollen grains lands at the stigma, there is what we call germination. They germinate. Now they germinate to form what? They germinate to form what we call a pollen tube. The pollen grain germinate to form what we call a pollen tube. Now what occurs after germination? What occurs after germination is the growth of now the pollen tube through the style. The pollen tube grows through the style down until it reaches the ovary. Now the pollen tube grows down until it reaches the ovary. Now after the pollen tube reaches the ovary, what happens now? The pollen tube breaks so that now the pollen grain inside the pollen tube meets or unites with now the ovules which is inside this ovary. That is how we discuss about fertilization. Now in fertilization, the most important thing you have to understand, number one, is the germination of the pollen tube which occurs at the stigma, the growth of the pollen tube which occurs in the style, and also now the fertilization that occurs in the ovary. Now after that, you have to understand now, after fertilization, what forms the seeds and what grows to form fruits. Now the ovules that are inside the ovary, they grow now to form the seeds. But now the ovary itself, it grows to form what we call the fruits. Now the part of the fruit that we eat is what we call the fruit, that is the ovary. But the seeds now which is inside the fruit, for example in a mango plant, this is a mango seed. Now this mango seed, I have removed the external parts. We eat the flesh part of it, which is now the fruit, but now inside here, this one is what we call now the seed. Now when we move to now the parts of the seed, after understanding how the seed grows, we move to the parts. The seeds have different parts. Now these seeds are divided into two. We classify them as the monocot seeds and also the dicot seeds. And in our introduction we talked about dicots and monocots, and you understand it better. Now the parts, we are going to start with the parts of the dicot seeds. Dicot seeds is made of, number one, it's made of the tester, and the tester is the external part. For example, this is a bean seed. Now this external part, which is colored, the colored part of this seed is what we call the tester. the colored part of the seed is what called the tester. Now when we move to this part here, which attaches itself now to the pod, is what we call the helium or the seed scar. We have a part here at the top which is very tiny, that is called now the micropyle. Now after the micropyle, when you remove the tester, when you remove the colored part, you will meet a part that is not colored. It is most of the time it's white in color. It's called the cotyledon. Now those are the parts of the dicot plant or the dicot seeds. Now we move to the function of these parts. What is the function of the tester, the micropyle, the helium, and the cotyledons? Number one, we start by the tester. The function of the tester, because it's the external part, it protects the inner part of these seeds. That's why now you will get it's very smooth, so that when you touch it, you cannot touch the internal part. So the tester protects the inner parts of the seed. Number two, we move to the micropyle, which now allows water and air to enter into these seeds. Now water enters into the seeds through the parts here, which is small part here, that it allows water and entry of water and air. Now during germination, we are going to discuss about it. During germination, the radicle, which is now grows to the during germination, it grows through these micropiles. The micropile, number one, allows growth of the radicle. Number two, it allows entry of water and air. We move to the third part, which is the helium. Now the helium, it attaches itself to the pods. This part here, the smaller part in the seed of a bean seed, is what we call the helium. Now this one attaches itself to the pod. When you open the seed of the pod, you will get that part here, which is called the helium. We move to the last part, which is the cotyledons. Now in dicots, they store their food in the cotyledons. So the cotyledon is used for the storage of food. Now when you open the helium, the tester, you will meet the cotyledons. Now there, that is the storage of food. That is all about the parts of the dicots. We move very fast to the parts of the monocots. Now a monocot, example of the monocot seed is the maize seed. Now this is the maize seed. Now this maize seed has different parts. Number one, it has the part up here, which is called the style scar. Then it has the second part here, which is called the cotyledons. Now the cotyledons is the part that makes most of it's oily. It has oils. Now it is made of the part here called the plumule, and the down part here is called the radicle. Now when you move to this part here, which attaches it to the maize cobs, you will get that part is called the fruit scar. It's called the fruit scar. Now when you open this maize seed to remove this white color, the tester, you will meet what we call the endosperm. So it has style scar, it has cotyledons, plumule, radicle, fruit scar, and lastly the endosperm. What are the function of those parts? Now the function of these parts, number one, we talk of the style scar. This is just the style remains. We talked about the style during the formation of the fertilization. We talked about the style. Now when fertilization occurs, we have that part that remains on the maize seed, the upper part of it, which forms now the style scar. When you move to number two is the endosperm. Now the endosperm is where now the storage of food occurs. Now we said in dicots, the food is stored in the cotyledons. But now when you come to monocots, food is stored in the endosperm. We will come to discuss the differences between monocot seeds and dicot seeds. Now the endosperm, the food is stored in the endosperm, that is in monocots. We move to the third part, which is now the radicle. Now in these monocots, the radicle grows to form what we call the roots. Radicle grows to form the roots, but now the plumule, which is the part that is the upper part of this seed, it forms what we call now the shoot. Now this shoot is just the young leaves that grows immediately after fertilization. Now the radicle grows to shoot, and then plumule grows to form shoots. We move to cotyledons. Now the function of the cotyledons in the monocots is different from those in the dicots. In dicots, it's just the general storage of food, but here it's now specific. It says storage of food, but now only for those parts that are germinate to form the shoot and the root. Now this part here, This part is what we call now the cotyledons. This cotyledons, it stores food for the growth of the plumule and the radicle. That's why we say now it is just storage of food for germinating parts. Now we move to the differences between now the dicot seed and the monocot seed. Now the differences, number one, we talk of the dicots, monocots, they have one cotyledons. When you open this, you'll just get one cotyledons. But when you open up this the bean seed, you will get that now it has two cotyledons. Let me use this of a mango plant, which is just the same as the bean plants. Now when you open this, this is the first cotyledons and this is the second cotyledons. That is, in dicots it has two cotyledons, while in monocots they have one cotyledons. We move to the second difference. We discussed under the roots. We said monocots, they have fibrous roots, and we gave an example of fibrous roots, those that grows from the base, while the dicots, they have what we call the taproot. That is the main roots, then it has some lateral roots. Then we move to monocots. They have, it's mainly made of cereals. We talked examples of cereals where we said we have the maize, we have maize plants, we have wheat, we have sorghum. Those are examples of cereals that fall under monocots. Dicots is made of mainly legumes. It's made of mainly legumes, and example of legumes we said beans, peas, groundnuts, and other examples. Then when you move to the fourth difference, this one, the leaves of these monocot plants, it is parallel. It has a parallel venation. We have that parallel venation, and the parallel venation, this is how parallel venation occurs. Now you have a maize plant, that's the leaf of the maize plant. Now parallel venation, we say those are the venation, they are straight from the base moving upward. Those are the parallel venation. When we talk of the net venation, that is in dicots, the net venation, that is a broad leaf like that, but it has now the main, that is the midrib, but it has some branches. This now means this is the net venation. So monocots, they have parallel venation, while the dicots have what we call the net venation. When we move to the fifth difference, in monocots, food is stored in endosperm. In monocot, food is stored in endosperm, we have discussed it, while in dicots, food is stored in cotyledons, we have discussed it. Now directly, we move to the another subtopic, which is now the germination of these seeds. After understanding the parts of the seed, the parts that grows into the plumule or the shoot, and the part that grows into the roots, we now move to now how does germination occur and what is germination. Now germination, this is just the process which now helps the seeds to grow into a new plant. This new plant is called a seedling. The new plant that grows is what we call a seedling. Now if somebody asks you to discuss a definition of the germination, you just say is the process by which the seed grows into a new plant, and that is, the new plant is called seedling. Now what are the conditions necessary for germination? As we discussed conditions necessary for pollination, the agents of pollination, we now have to discuss also the conditions necessary for germination. Now for germination to occur, it requires three main things. Number one, it requires what we call oxygen, that is air. Number two, it requires water, and lastly, it requires a suitable temperature. We talk of suitable temperature because when the temperatures are very low, germination cannot occur, and when the temperatures are very high, above optimum, the germination cannot occur, they cannot occur. Therefore we talk of a successful or a suitable temperature. Water and air, it doesn't have any minimum, but temperature, we'll discuss it as we move on. Now they can draw for you an investigation, they can draw for you an examination, an investigation carried out either to check on the successful of germination. Number one, how can you investigate that oxygen is a requirement for germination? How is oxygen as a requirement of germination? Number one, you need the following. Number one, you need two containers, and in the two containers, you have to boil water. You put in boiled water in each container. Then one container, you cover it, the upper part of it, you cover it with oil. Now the two container, name them A and B, and now inside it you have seeds, that is either bean seed or maize seed. Now you observe after maybe 1 week, you come and observe germination, which seed will germinate and which one will not germinate. Now in container A we have air, we have allowed the entry of air, and in container B we have oil. This oil prevents the entry of oxygen or the entry of air into this container here. Now because here we have air and everything, in container A you will find out that the observation will be, number one, there will be successful germination. This seed will germinate. But in container B there will be no germination. That's the seed will not germinate because we did not allow the entry of oxygen. That is all about how to investigate air or oxygen. We move to how now you can investigate that water is a requirement for germination. For a successful germination, we have to have a water which is now has a requirement temperature. You cannot just have water, but water that has a requirement temperature. That's why most of the time we talk of moisture. Most of the time we use moisture. Moisture is used in terms of water. Now, here you have also two containers. Name them, you can name them C and D. Now in container C you have a dry cotton wool, and in container B we have a wet cotton wool. This wet cotton wool, it means it has water or it has moisture. Now after some 1 week you come and check on these two containers. You will find out that in container C we will not have germination. Germination will not occur in this container because there is no moisture here. This is a dry cotton wool, and this is a wet cotton wool. There is presence of water. So observation, germination will occur in container D, but germination will not occur in container C. Reason, here there is no water and here we have water. So successful germination requires water. Now how can we investigate that now temperature or warmth is a requirement for germination? This is warmth, which is also called temperature, but temperature we say suitable temperature. Now how can we investigate? Number one, you also need two containers. In the first container you have just water. You have water, it is open, meaning there is entry of air. We have water, we have the seed. Number two, we have here the second container. Yes, it has water, but we have ice cube. Then we have the seed. Now what will happen? In this container, after 1 week, we will observe that in container E, this is container E and this is container F. In container E, we will have a successful germination because water is at a required temperature or a required warmth. Then in this container here, which is now an ice cube, the ice cubes has a very low temperature. They have very low temperature. Now if they have low temperature, it means that temperature will not favor the germination of this seed. So with the here observation will be in container C we will have a successful germination. That the seed will germinate. While in container F we will not have a successful germination because of the presence of the ice cube, which decreases the temperature to a point where it will not have to support germination. Now what happens to the cells when there is low temperature? When you move to second level, you'll discuss the effects of low temperature where it denatures the cells of these plants. Now when you talk of denaturing, that's destroying the cells, hence no germination. Now we move to another subtopic under germination. We talk of now the stages of germination. For germination to occur, it undergoes some stages, 1, 2, 3, and up to number 5. And these stages, you must understand them the way it occurs one by one, because you may be given in an examination and they ask you now to arrange the stages the way it occurs during germination. Now, these stages occurs this way. Number 1, we have the absorption of water. This absorption of water, we said it occurs in the micropyle. We discussed during germination. Here, we said the micropyle, it allows entry of water and also it allows entry of air. Now the first thing, water enters into the seed through a micropyle. After the entry of water into the seed, this seeds, number 2, the seed swells. After water has entered into these seeds, for example, this is just a smaller seed, but if you take this seed and put it into water for about 2 days, you'll get this seed has swollen. it has become big or swollen. Now if the seed has swollen, what happened? There is this bursting of the tester. The outer part, which is the tester, now bursts or open. The tester bursts or open. That is the number 2 that occurs. Number 3, after the tester bursting or opening, there is that radicle now comes out. The radicle, this one, there is the first step that crosses the radicle. The radicle, we said, it grows into to produce the roots. So these are the roots, which are the radicle. Now after the production or the growth of the radicle into roots, there is the 4th step, which is now the cotyledons. The cotyledons of this plant, for example, this is a seed which is a dicot. Now this dicot, we said, it has 2 cotyledons. This cotyledons, now the side, the one cotyledon on this side and another one on this side, it opens up. When these cotyledons open up, there is the 4th that allows now the plumule to grow. This plumule now grows because of that opening of the cotyledons. That's now the growth of the plumule. And we said the plumule grows to produce what we call a shoot. And shoot, I said, is just young leaves of that plant. Now the last stage that occurs during germination is what we call now the growth of the new plant. And new plant, we said, is what we call the seedling. Now a new plant is formed. This new plant is what we call the seedling. Now somebody can ask you, the combination of the plumule and the radicle is called, now if somebody asks you that a combination of the plumule and radicle is what it forms, what we call an embryo. This one forms what we call an embryo. So plumule and radicle forms what we call an embryo. Now summary, today we have discussed fertilization. You have to understand the definition of fertilization in plants, where it occurs, the parts that are involved in fertilization, and also we have discussed the parts of the seed, that's the dicot seed, monocot seeds, and their functions. We have discussed the differences between the monocot seed and the dicot seed. You have to understand it. We have discussed the germination, definition of germination, and also the requirement, the condition. You have to understand the 3 conditions. Very important. You can be asked which one of the following is not a requirement for germination. So you must be well versed with the investigation part of it. You have to carry out investigation at personal level so that you can understand it better. Then lastly, we have discussed the stages of germination that occurs, that is first to the last stage. So that is all about today's science lesson. Let's meet next time, inshallah.